I want to remind you what we have finished with on Thursday. We're talking about malaria and specifically the symptoms and the distribution of the disease in the world. Okay, so South America, Southeast Asia, Central Africa, the huge part of Africa, and that the disease is transmitted by the bite of mosquito. Okay, so mosquito takes a blood meal from infected person, then transfers that blood sample into the uninfected person, transmitting the plasmodium. Talked about symptoms, chills, fever, shaking, chills, and few things that we just didn't have time to mention. First of all, a little bit more elaboration on the symptoms. First of all, symptoms as we mentioned them, chills, fever, malaise, nausea, they sound horrible enough, but they don't sound deadly, do they? However, in small children, what happens? Plasmodium can travel to the brain and get into the capillaries of the cerebral circulation and significantly deprive the brain from oxygen by impeding the blood flow. And when you decrease the oxygen supply to the brain, what's going to happen to the person? Initially. Huh? Well, it's not complete cessation of the blood flow. Blood still gets to the brain, but oxygen delivered to the brain, it's like chronic brain ischemia. You lose consciousness. Now, when you have lost consciousness for days and weeks, we usually call it coma. So it's called cerebral malaria. Kids very quickly slip into coma, and especially in children, cerebral malaria almost inevitably ends in death. That's number one. Number two, just a little aside on mosquitoes. What do mosquitoes eat? That's my question to you. Blood, huh? No, not other insects. They don't eat blood. In fact, the mosquitoes that take a blood meal, that bite you, which sex are they? Females. What about males? What do they drink? Like beer? That's it? No food? What do they eat? They eat sap. Like the plant juices. Both males and females. Females eat sap as well. What do they need blood meal for? Say again? That's good. Reproduction. They need source of protein for development of eggs. Okay, so that's why one of the directions in mosquito control is to either make them not only genetically sterile, but also make them able to. There's something like weird thing, so they not making them sterile, they're making them genetically modified, so their offspring is sterile. So they want to spread this gene through the population. It's some very, very complicated genetics involved, which I am not really familiar with. But the idea is to eventually eradicate mosquitoes. Do you think it's a good idea? Seriously, I would be the first person to say I have no regret in getting rid of mosquitoes. But the problem is, it's one of the major food sources for amphibians. So we might as well just get rid of frogs and toads as well. It's actually a big deal. Another direction generally is, so in order to transmit malaria, how many blood meals does one mosquito have to take? 2, yes. One from infected, one from uninfected. So there is an idea if somehow the behavior genetically can be modified, so the mosquito takes only one blood meal, then it helps. You have to understand, though, that this treatment is somewhat altruistic. Does that make sense? Because it means that if somebody is sick, it's not going to help this person. There's going to be no transmission, but those who are sick, they're already sick. Okay? Now, the life cycle, I know, this is scary. And I want you to walk through the steps because I think this picture is a little overloaded schematically. But since it's a copyright-free use, it's convenient. So, okay, we've got the person in the center, okay? And we've got a mosquito right here that takes a blood meal and injects so-called sporozoites. And those sporozoites first go to the liver. That is why one of the characteristic symptoms of malaria is hepatomegaly, increased liver, okay, in size. And there are also often elevated liver enzymes that show the liver damage, right? Now, in the liver, Plasmodium goes into the stage of merozoite. Just different stage, sporozoite, merozoite. And then, listen up. Every so often, bless you, every so often, merozoites are released in the blood. Guess what? Plasmodium falciparum releases merozoites every 3 days. Every 3 days you experience fever, chills, and bouts of malaria. So bouts of malaria are directly linked to the type of Plasmodium and to its life cycle. As far as I remember, Plasmodium malariae releases merozoites every 2 weeks. Guess what? Every 2 weeks you're going to have symptoms of malaria. Does that make sense? Okay? Does that make sense? So this is something I really want you to process here. The symptoms of malaria, the frequency of symptoms, or the course of disease is first thing that defines it is the type of Plasmodium and its life cycle, essentially. Okay? Now, in the blood, Plasmodium gets into the red blood cell and multiplies in the red blood cell. Alright? This is how you diagnose. Okay? This is how you diagnose. That's a sky zone, that's a ring form of Plasmodium right here. You can see them on the microscope, under the microscope. That's actually sort of a convenient thing for countries like Africa, or countries in Africa and South America, because clinics may not have PCR machines or other sophisticated equipment, but local clinics, hospitals, they do have microscopes. So you just do the simple smear. There are actually 2 smears. There's a thick and a thin, and I don't remember the diagnostic role of each of them. But essentially, you do the blood smear without any staining. You just see the Plasmodium, which is convenient. Now, interestingly enough, when Plasmodium is in the blood, the red blood cell, red blood cell, what is it filled with? Hemoglobin. And hemoglobin is what? Iron. And what else? No. What is the big part of hemoglobin? Globin is what? No, globin. It's a protein, yeah. It turns out heme and iron are toxic for Plasmodium. So Plasmodium does a very interesting thing. It destroys globin part and uses it for it's a protein, it's a source of amino acids. And heme molecules are being stacked in the corner of red blood cell. Well, it's round, so there are no corners, but like on the side, because it's toxic. Okay? And then eventually this ring stage the merozoites in the ring stage. They develop in gametocytes. And guess what? Plasmodium, although it's a single cell protozoa, single cell organism, it has male and female gametocytes. These male and female gametocytes, they taken up by the mosquito, okay, and then they go through the fusion of pretty much like a sexual stage, okay, male gametocyte, female gametocyte, they fuse, form zygote, zygote forms sporozoites. I'm going to do the summary of the life cycle, but I want to ask you a little question. So sexual stage of development, sexual stage, in which organism does it happen? Sexual stage, in mosquito, okay? Now there's a definition in parasitology. Sexual stage occurs in definitive host. That kind of turns around our concept of humans being at the center of it all. Think about this. For Plasmodium, humans aren't definitive hosts. We just are meaning to develop and to reach the sexual stage of development in the mosquito. Mosquito is the main host. Mosquito is the definitive host. Humans, in this case, are going to be intermediate host. To sort of sum it up, mosquito injects sporozoites in the bloodstream. Sporozoites go, now this what I'm telling you, I may ask, sporozoites, not in any more details, sporozoites go into the liver. Okay, mosquito injects sporozoites. Sporozoites go into the liver, become merozoites. Merozoites from the liver enter the bloodstream every so often and infect red blood cells. In the red blood cells, merozoites form gametes, or gametocytes, sorry. Gametocytes are sexual. Gametocytes are taken during the blood meal by mosquito, which is definitive host. Gametocytes in the mosquito form zygote. Zygote produces new sporozoites. Sporozoites are again injected into the human during the blood meal. That's it. Does that make sense? This is the level of knowledge for the life cycle that you have to stay at. Don't go deeper. That's more than enough. Do you want the sad story? It's a really sad story. New York family, parents are divorced. The father, I cut it down. I also am not very good at telling sad stories, so that people will start crying, you know. I've heard it in much more expressive version, okay. I almost cried. So the father marries again. There's a woman, and stepmother has troubles establishing the connection with her stepdaughter. So she finally, and they're good people, just, you know, it sometimes happens between the adults and the kids. And she decides to do something, you know, to create a bond with the stepdaughter. And she tells her, let's do a secret trip. It's a pretty wealthy family. And she said, we're going to go to Kenya for a trip, secret trip for a couple of weeks. We're going to go on safari, you know, look at a bunch of animals and all of that. So they decide to go, and they go on that trip, spend 2 wonderful weeks in Kenya. And then the daughter comes back, mother comes back, and daughter starts to complain about the fever and back pain. That's a very characteristic symptom of malaria, back pain, lower back pain. And she doesn't sleep well, she doesn't feel well, but the mother says it's a secret trip. So we're not going to tell anyone about it. And when the girl goes to the doctor asks her, have you been out of the country recently? And she says, no, I haven't. And she keeps it a secret. She's very good. She's a good girl. Okay, she keeps it a secret. It's a secret trip. So she goes, back and forth from being okay to being sick, okay, sick, and then obviously they didn't take any anti-malarial drugs, so she's infected with malaria. And she eventually sleeps into cerebral malaria. So she essentially sleeps in a coma. And she's brought in one of the hospitals in New York, and some physician decides to take a blood smear. And of course he finds malaria, a ton of them, plasmodium. And they start to treat her because she's in a coma. And I believe they treated her with quinidine. Quinidine has an interesting feature. I think it is quinidine. It can induce ventricular fibrillation in the heart. And it does. And the girl has ventricular fibrillation, pretty much. Her heart stops. And what doctors do when the heart stops before they get pads? Yeah, they do the compression, yes? Well, there's another thing. At this stage of malaria, it's not only hepatomegaly, it's also splenomegaly. So her spleen is enlarged. When they do CPR, they had no idea what's going on. They accidentally rupture the spleen. She dies in 2 hours because of the blood loss, which illustrates one saying by infectious disease physician Harold Brown. Patient history is the most valuable and the least trustworthy piece of information that you can get. Toxo, a fantastic protozoan. It's apicomplexa, and it's obligate parasite. Turns out it's so obligate, it can form cyst, but it doesn't form cyst outside of any organism. It's always in the living organism. Okay, it can be in a feces. Okay, but doesn't replicate outside. It can only replicate in living organism. So oocysts from the fecal matter can be eaten, okay, by rats or birds, okay, and they ingest feces. We're going to talk about this scheme in a little bit. I will make sense out of it, okay? So one way is birds and rodents get oocysts from the fecal contamination of water or food, and then cats, for instance, okay, they can eat rodents. So oocysts become the living trophozoites. They have a special name, tachyzoites, but trophozoites, okay? And we'll stop at that. So now imagine you have a cat that is infected with Toxoplasma gondii. Cat will defecate on the litter, and feces of the cat that was infected by Toxoplasma gondii, because cat ate the mouse that was infected with Toxoplasma gondii, those feces will be contaminated. Now you handle feces, you get this oocysts on your hands, inevitably they get into your mouth or in your food, and then you consume the oocysts, you have the infection, right? Does that make sense? So one way, I think this scheme is horribly overcomplicated. So think about this. One, it's a cycle. Rodent gets the cysts, develops the infection. Mouse, it's the rodent, develops the infection. Sheds cysts, humans get infected by handling cat litter. We got it? Second way, cattle and pork and pigs. They get oocysts from the environment, from contamination. Okay? Oocysts develop into the tachyzoites, and then we eat meat and beef and pork. Let's be honest, when you order or you cook a steak, okay, you grill a steak, how many of you eat well done? I don't see any hands. Okay, good job. Good job. For me, it tastes like a piece of rubber, honestly. So I don't care about, I want to die sick, not healthy. So I eat medium rare. Now, when you don't cook meat well, tachyzoites get into your digestive system and they get into the tissues, they can get into the muscles, they can get into the brain. And you know what's interesting? There are no symptoms. In immunocompetent individuals, there are absolutely no symptoms of toxoplasmosis. About a quarter of US population is infected. In the world, the estimation would be, if we would even say, okay, quarter of the world population is infected, think about this. This is goddamn 1.7 billion people, and the estimations are larger. They suggest that 2 to 2.5 billion people are infected with toxoplasma. Tell me about evolutionary success. In France and in Brazil, the percentage of population that is infected is at 80%. I'm talking about people, I'm not talking about cats. Any reason, any idea? Think about, okay, go ahead.
Well, like, you know, they eat a lot of, like, raw stuff and things like that, so
Have you,
killing the bacteria or the, you know.
absolutely. One of the most popular dishes in France is steak tartare, which is practically raw beef. I mean, it's as close to raw as possible. In Brazil, if you've been to a Brazilian restaurant, I believe there are a couple here, I went once. It's the meat lover's dream. They serve meat. Just a ton of it. So, and obviously people don't always request well done, so the rate of infection is huge. Okay? Now, who is at risk? Immunocompromised. Huge risk. Have you heard about the gentleman named Martin Shkreli? You haven't? Well, I think this person needs more publicity than he already got. Martin Shkreli was quite successful Wall Street finance guy who decided to employ a new scheme that was caused by a loophole in the US laws. He bought a patent for the drug that is used to treat toxoplasmosis and other parasitic infections in AIDS patients. And it was cheap because patent expired. So everybody could make it and everybody did. I don't know how legal system works, but he bought a patent and jacked up the price of one pill to something like $700 or $800. So yearly treatment, and it's dirt cheap. Yearly treatment for toxoplasmosis became something in the ballpark of $30,000 or more. Okay. Of course, it caused a massive outcry in the AIDS patients community, in medical community, in all patient advocate communities and whatnot. And US regulatory organs were not happy about it either because they realized they fucked up massively. But it was all legal. He didn't break any law. So they couldn't do anything. except for one thing. Securities and Exchange Commission, SEC, immediately when he did that, they started to investigate his financial deals and found him, in 2 months, they found him guilty of fraud with all the evidence, and he was imprisoned. Which, I love the story. It's like, don't trouble troubles until troubles trouble you. Don't try to outsmart the system, because he was essentially an asshole, honestly, and he got it. Now, interesting feature, kids, newborns. Congenital toxoplasmosis is extremely severe. CNS damage, so that's why for pregnant women, it is not recommended to handle cat litter, which is one of the major sources of toxoplasmosis, and also not recommended to eat undercooked beef or pork. Now, interesting feature of toxoplasma is how rodents get eaten by cats. Turns out that rats and mice that are infected with toxoplasmosis, and its neurotropic infection goes in the brain, they stop fear the cat's urine. That's the way they know the cat is around. They sense the smell of the cat's urine. When they're infected with toxoplasmosis, they do not sense that smell, and they can come like face to face with a cat. And of course, cat is going to eat them. Essentially, the parasite modifies the behavior of the animal to facilitate its own transmission. There are reports that toxoplasma may be involved in development of schizophrenia in humans. This is generally not accepted by the scientists that work on toxoplasma. Those are epidemiological reports, because French, as crazy as they are, they definitely don't have elevated number of schizophrenia in population. Okay, so it's kind of, whether it can influence human psychiatry, we don't know, but in rats and mice, it definitely can. These guys take a special place in my heart, not literally, but figuratively. Helminths. These are the only multicellular organisms that we talk about in a course of microbiology. There are 3 types. Nematodes, or roundworms, trematodes, or flatworms. They are also called flukes. They flat, and not just flat, they are non-segmented. And cestodes, or tapeworms, which almost always segmented. Okay? So you need to know the classification. I will ask. Understand the shape, like flukes, flat, non-segmented, nematodes, cylindrical body, long body. Other major differences between them: nematodes have the body cavity. If you would take a simplest nematode, Caenorhabditis elegans, which is not parasitic helminth, it's a tiny, tiny worm, about 1 millimeter long maybe, that feeds on bacteria. It has neurons, it has digestive tract. Actually, the entire map of Caenorhabditis elegans neurons have been established. So we know which neuron communicates with which. It's something like 900 cells, something in that ballpark. Flatworms, whether those are trematodes or cestodes, they do not have body cavity. And roundworms are sexual, so they have males and females. Flatworms are hermaphrodites, except for one, but I'm not going to focus on it too much. Okay, I'm not going to ask you the tricky question about the schistosomes have sexual stage, but that's an exception. So how do we treat against helminths? What's the problem? Again, what is the problem with the drugs that target helminths? We've already ran into this with... Yes, why? Exactly. They are eukaryotes and we are eukaryotes. So these drugs are pretty toxic. And often, you have to very carefully consider the course of treatment, because deworming long-term treatment may have quite a detrimental effect on the health of the patient. Another thing, if you think about treating the adult worm, they may reproduce really slowly and they don't die. That's actually an interesting feature. Some of those worms are technically immortal. If you take a tapeworm, put it in the mouse, it will live in the mouse for years. When the mouse is about to die, you can take the tapeworm, same tapeworm, put it in a new mouse, it will live in the new mouse. By transferring it from one mouse to another, it never ends. They're primitive, of course, but technically they can live for, many of them can live for years and years, humans or in animals. So, even if you block reproduction, you still have an adult worm which is active. Okay, so you have to somehow target an adult worm itself. Does that make sense? So there are pretty much two options that are used mostly. It's immobilization of the worm or disintegration of the worm. And there are two big classes of drugs. One is mebendazole. You don't have to know the formula, of course, but next time you go to Petco, PetSmart, or whatever pet store, you have pets, many of you have pets. If you may see little boxes for deworming, anti-flatworm treatment or tapeworm treatment. This time, grab this box and read the content. It's going to be either mebendazole or thiabendazole. Those drugs were developed, I think, by Merck. And what they do, they inhibit synthesis of microtubules. Worm, as any living creature, is multicellular organism. Cells in the worm die. Does that make sense? So new cells have to be generated. And cell growth and cell division involves microtubules. Microtubules have to be synthesized to make new cells or for cell division. When you inhibit microtubule synthesis, you inhibit cell division, worm slowly, quote-unquote, decays. Okay? They're very effective, actually. Another great drug is ivermectin. Ivermectin developer William Campbell received the Nobel Prize in Physiology and Medicine in 2015. This drug is, I heard a great name, dihydroxychicken wire. So what it does, it stimulates chloride channels in the cell. Speaking generally, now you have to recall the Anatomy and Physiology 1. In general, chloride concentration in the cell and outside of the cell. Where is it higher? Generally. Yes, it's higher outside. You can say chloride, sodium, calcium outside, potassium inside. Okay, so chloride is higher outside. So when ivermectin interacts with those channels, it opens them. So where would chloride go if it's higher out? Huh? Inside the cell. What's going to happen to the membrane potential of the cell when negatively charged ion enters the cell? It's going to become more negative. How do we call this in terms of polarization? Hyperpolarization. Hyperpolarization, what is it? Does it make cell more excitable or less excitable? When it becomes even more negative, it becomes less excitable. So remember, that's the action potential. So in order to reach action potential, you gotta bring positive charges in instead of bring negative. In case of hyperpolarization, cells cannot be excited. If those cells are neurons, transmission of the signal stops, worm is paralyzed. Now, this is one thing. It does not kill the worm. It doesn't, but it paralyzes it. So it doesn't move anywhere. It cannot do anything. It cannot replicate. It cannot move. It cannot do anything. Does that make sense? Let me see. Okay. I'm going to do a little story about ivermectin. I don't talk about many worms here, I think. Yeah, I don't talk about many worms. But there is a disease in Africa called river blindness. I think I mentioned it before. It's the disease transmitted by black flies, caused by the worm Onchocerca volvulus. It's a round worm. Since it's not in a PowerPoint, you don't have to worry about it. So this round worm gets under the skin first, then grows. Some worms can be up to 6 feet long. They're really long, but rather tiny, and they end up in the eye, and they cross the eye, and they can be diagnosed by somebody sees it crawls across the eye, and it leads to blindness. This disease was having a pretty big impact on the villages in Africa, and Jimmy Carter, when he stopped being president, he founded the Carter Foundation. One of the purposes of Carter Foundation was to get rid of river blindness. So what he did, he talked to Merck executives. Merck was actually producing ivermectin, and convinced them to bring it to African market for free. Well, I mean, sell it for 0 cents. $0, 0 cents. Carter Foundation was taking responsibility of shipping and logistics. And guess what? Onchocerciasis is not a severe problem anymore. You can say, well, wait a minute. Merck, last time we checked, Merck used to be a commercial company. They're making money. Where are they making money from? No. They're selling ivermectin. To whom? To you. You're buying it to treat heartworm in dogs, because it's also nematode and things like heartguard. That's ivermectin. Okay. So just several helminths that we're going to talk about. First one is Tinea. Love that guy. It's wonderful. Tinea, there's several Tinea. Tinea solium and Tinea saginata are most common. The cycle is simple. Eggs or fragments of the worm, so-called proglottids. Okay, proglottids. They are on the soil, in the feces essentially. And then cows and pigs They eat the respective eggs. Cows, they are infected with Taenia aginata, pigs with Taenia solium, and those eggs develop into the larval, kind of sort of larval stage, oncospheres. Okay, it's essentially a larva. Does that make sense? Now, interesting thing about these larvae is that they start to develop, eggs hatch and develop into larvae in the intestine, and then larvae migrate from intestine into the surrounding tissues, mostly muscles. Okay, remember, this is still animals. So in this case, cattle is going to be intermediate host. Now, what do we do with pigs and cows? Which part of pigs and cows we usually eat? Muscle. Well, tongue is really good. And then when you eat muscle, where you have larvae, you essentially ingest that larva. Larva in the intestine develops into the adult worm that can be several feet long. It attaches using so-called scolex. You can see the head of the worm. Head is called scolex. So on the scolex, you have suckers and you have hooklets. Okay, so it sucks itself like the holder in your bathroom on the wall. You have the thing with those suckers. That's the same. It attaches itself to the wall of intestine and using hooklets grabs it better and just hangs off the wall of intestine. And shape-wise, it's like a really long ribbon that contains multiple fragments called proglottids, like a chain of proglottids. What does it eat? What does it eat? What does the flatworm eat? Does it eat you? No, it doesn't. Exactly. You share the meal. Does it eat a lot? It's a very primitive organism which just hangs from the wall of intestine, so it doesn't. So the whole idea of getting infested with a tapeworm to lose weight doesn't really work. It's not going to happen. I mean, you cannot possibly eat that many flatworms. What symptoms of flatworm infestation are usually none. Maybe a little bit of discomfort, abdominal discomfort. There were cases when during colonoscopy, 3 flatworms were found in the intestine of a woman and she had no idea. She didn't feel anything. Okay. Now, the flatworms are there. So what do they do next? Those tapeworms, sorry. They shed either eggs or they can also shed proglottids. And actually, people who are infested with flatworm can see proglottids on their underwear. So they can pretty much leave the anal orifice without defecation. So that's often how people diagnose themselves. They see little, and they depend. Some flatworms are smaller. Some species of Hymenolipis nana, which is the fish, one of the fish flatworms, can be, I don't know, like a few centimeters. So proglottids are really small. The Taenia solium or Diphyllobothrium latum, that's another fish tapeworm, they can reach land of 40, 50 feet. So proglottids are pretty considerable size. Okay, and of course, so we shed eggs and we shed proglottids. And then these eggs and proglottids, which we shed in ginormous numbers, honestly. We talk about thousands of eggs a day. They end up everywhere, including soil where, animals will eat them, and the cycle goes on and on and on. Does that make sense? So animals, like cattle and pigs, they will eat eggs. Eggs will develop into the larva, that's intermediate host. Then we eat larva that resides in the muscle of the pigs. We actually are definitive host. Now let me ask you a little question. We shed eggs. Can we eat them? Potentially. Can we eat eggs of the Tinea solium or Tinea saginata? What do you think? If we shed them like everywhere, hands, food, can we? Sure. I mean, why not? Contamination, fecal contaminations everywhere. What's going to happen to them if they get not in the designated intermediate host, but in humans? What do you think? Are they going to hatch? Well, imagine you are a Helminth egg. Would you? Well, I would check the environment first, and if the environment seems similar, seems right, then I would definitely hatch. Well, it turns out that Tinea saginata rarely does it, but Tinea solium, the pork tapeworm, does it a lot. Because pigs and humans are quite immunologically similar. Does that make sense so far? Now, before we move on to what happens next, let me tell you the story. New York. I don't remember which year was that. I think it was 20th century. New York. And in few Jewish families, Orthodox Jewish families, Orthodox Jewish families, people fall ill with the disease that is called neurocysticercosis, which is larva of tapeworm migrated into the brain. Remember we talked about eggs hatching in the intestine, right? When eggs hatch into intestine, they become larvae, and larva migrates from the intestines into the surrounding tissue, muscles, and the brain. So larva gets into the brain, and look, if you have a larva getting into the brain, your brain is not going to function as it used to be, right? So those people have symptoms of neurocysticercosis, which ranges from headaches to confusion to behavioral changes to seizures and to epilepsy. Okay? And they brought in the hospital, and few of them die, and on autopsy they diagnosed with neurocysticercosis. And guess which species is that? It's Tinea solium, pork tapeworm. Wealthy Orthodox Jewish families, members of those families are infected with pork tapeworm. I don't know about you, but it doesn't add up in my mind. Didn't add up in the minds of CDC officials. They started to investigate the case. Well, they excluded Jews. It's just not really feasible. They started to look at everyone who lives in the house. And they started to swab for eggs. Not people, surfaces like window sills and tables. They were covered with eggs. Dust on the window sills were pretty much a mixture of dust particles and Helminth eggs. They were everywhere, on every surface in the house, so no wonder those people got infected. But where did those eggs come from? Because where do eggs come from? I'm showing you here, human rear end, yes, human feces. So there must be someone in the household who carries an adult tapeworm, which in turn produces eggs. And remember, three or four families. They're not related, they're not in the same household, they're friends though, but not related. They started to look at the house stuff. Turns out, one family hired house maiden from Mexico, just like a year ago. She was really good at her job, and they recommended her to the friends, and she actually kind of called her sisters and relatives. So they got permission for them to travel to New York and work as housemaiden for other houses. Pork tapeworm is like common cold in Mexico. It's absolutely common. Those women were all from the same village. They were all carrying tapeworm, and they were all shedding eggs. They shed so many that when they started to swab their skin, they found eggs in places like armpits, just laying there. So they not deliberately, but just accidentally, inoculated those people with that pretty unpleasant disease. This map actually shows you the distribution of Taenia solium in the world. Now, to give you an idea, you understand, the darker the color, the more transmission you have. I love to look at these maps because it's inquisitive and very educational to connect the disease and the social environment. Okay? So dark colors. You see them. Can you see them? So it's India, China, and Indochina. It's Africa. It's South America and Mexico. Well, pretty much South and Central America. What can you say about those countries? What's common about them? Huh? They're all developing countries. So what about sanitation? Poor. Very poor sanitation. So people, and do you think there is a USDA, United States Department of Agriculture, or FDA level of control? No. US controls every piece, well, not every piece, it's impossible, but they do frequent controls of pork that comes imported into the United States. So it's all clean. Does that make sense? In those countries, nobody cares. It's number one. Now, look, this is the northern Africa. Countries like Libya, Morocco, Egypt, Tunisia, they're still developing. There's practically no transmission. The entire Arabian Peninsula, Iran, Pakistan, Kazakhstan. Why? Exactly. They don't eat pork. Their religion bans them from eating pork. Does that make sense? So you can see that link. Does that make sense? It's for me. It's really interesting. Now, if you want to see sort of a movie, first episode of House MD, Neurocysticercosis. They actually portrayed pretty well. Well, as for any episode of House MD, I don't think there is any disease that can develop and kill you in 48 hours, as it usually does in those episodes. Other than that, they often pretty accurate, especially infectious diseases. Can be treated. So it's treated with mebendazole or thiabendazole. Often steroids are warranted. And this is the story. Think about this. You have a worm in the head. Well, larva in the head, in the brain. Your immune system will try to recognize and kill it. So in order not to get killed, larvae have very good immune suppressing molecules when it's alive. When you start treating with thiabendazole, it dies. No more immune suppression, you have a massive inflammation. Your immune system tries to destroy it. So patients may develop huge brain edema, so they have to be treated with steroids at the same time. Does that make sense? Schistosoma. There are many species: Schistosoma monsoni, Schistosoma hematobium. Never mind. It's a trematode. It's a fluke, and it's a. You can see them. Those wonderful worms. It's called also a blood fluke. Okay. Eggs present in mostly in feces, also present in urine. And eggs have several stages of development after hatching. And essentially, when they hatch, the first stage, Myricedia stage, you don't have to know that Myricedia infects the snails. And then snails release so-called cercariae into the water, and they swim in the water. Then people go swimming. Cercariae have an interesting feature. They can penetrate unbroken skin. Skin that is not damaged. If you would be a tiny little beast, how would you penetrate an unbroken skin? Where would you go? Which structure in the skin would you go? They're not burrowing through the skin. They use something that's already there. Huh? Hair follicle, yes. They go into the hair follicle and they penetrate by the hair follicle, right? And they end up in the blood vessels. And people with schistosomiasis develop rather non-specific symptoms initially, like diarrhea and abdominal pain and whatnot, but hepatomegaly, splenomegaly. Now, they impair the circulation in the liver. So people develop portal and pulmonary hypertension. And as you may remember, pulmonary hypertension is untreatable, especially idiopathic. Well, it's not idiopathic pulmonary hypertension, it's infectious, but it damages the blood vessels in the lungs, and it's almost inevitably leads to the heart failure, congestive heart failure. Okay. Another thing that they cause, inflammation of the bladder. As far as I remember, that's Schistosoma hematobium that ends up in the urinary bladder, causes inflammation of the bladder epithelium, and chronic inflammation of the bladder epithelium may lead to bladder cancer. And it's very common complication of untreated schistosomiasis. Another interesting thing, there's an evidence, well, there are records of men having menses in ancient Egypt. By menses, of course, the people who wrote this meant blood and urine. Okay? Hematuria. They thought that it was sort of a man's version of menses. Now, the main hypothesis, that was simply schistosomiasis that ancient Egyptians acquired after swimming in the river Nile. Is there a way to prevent it? Infection with schistosomiasis. Don't swim. There's another. It's not just swimming. Think about, again, look at the distribution. It's mostly Africa. So imagine the African kid who jumps into the Lake Victoria and swims in the Lake Victoria. If the kid survives swimming in Lake Victoria and avoids all the crocodiles, jumps out, does this kid have a towel? Africa. Probably not. Now, when you get out of the water and you don't have a towel and you dry out, how does your skin feel? I don't know, the kind of tense, when the water dries, the skin starts, it feels like it's being pulled a little bit. This drying moment is when Schistosoma monsono, Schistosoma, whatever Schistosoma species, that's the moment when they enter the hair follicle. So essentially, if you swim in the Lake Victoria, when you get in the shore, dry yourself with a towel. That's almost 100% prevents the infection. And of course, if you see symptoms, it's easily treated, really easily treated. So it's not going to kill you in a few days. Praziquantel, it's the drug of choice. That make sense? So another way is, of course, to get rid of snails, but it's not really possible. Fasciola hepatica, a great, interesting parasite. That's the liver fluke. That's a fluke. It's pretty large. I cut out the ruler located next to it in the picture, so it's kind of edited picture. To give you a perspective of how big it is, it's about 1 centimeter or, well, ½ inch long. It can reach ½ inch. So it can be visible with naked eye. It's pretty small. So eggs are eaten by the snail. In fact, snail, okay? It's one intermediate host, okay? And then snails release cercariae. Again, and then cercariae end up, that's intermediate host again, intermediate host. And then cercariae end up on the plants that are near water. It can be grass that animals eat, and then animals like sheep and cattle, they will pass the eggs and the feces and the cycle will repeat itself. Or it can be things like watercress, plants that require a lot of water for vegetation, and we eat them. And if those plants are contaminated with the cercariae, we develop the infection with the liver fluke. So adults migrate from the digestive tract into the biliary ducts of the liver. Remember the hepatopancreatic ampulla that delivers bile and pancreatic juice into the small intestine? That's how these flukes enter the common bile duct. And then by a common bile duct they reach hepatic bile duct and end up in the ducts of the liver, and then actually can burrow through the liver. And you can see on, I don't remember what they use, MRI or ultrasound, one of the imaging technologies, you can see the tracks. They burrow through the liver. Okay, essentially causes the hepatomegaly, liver inflammation. But what is more interesting, I think it's just absolutely brilliant from the standpoint of the snail, not the snail, the fluke, sorry. It also enters the bile ducts, the bile duct that leads to the gallbladder. And what does it eat? It eats the epithelium. Epithelium of the bile ducts. Okay, bless you. Now, unlike stomach, gastric epithelium or intestinal epithelium, epithelium of bile duct is not replaced very frequently. So, snails, flukes, stimulate epithelial cells. So think about this. Okay, think about this from the standpoint of a fluke. It's in the bile duct. It eats epithelial cells. Epithelial cells, as everything that's good, come to an end. So what do you need to keep on living in the bile duct? New epithelial cells. The fluke releases chemicals, releases mitogens that stimulate the division and growth of new epithelial cells. It's like a little farmer. It promotes the growth of epithelial cells so it has something to eat. Does that make sense? Does that make sense? Now, if something stimulates cells to divide, the more cells divide, the higher the probability of what? Huh? Mutation means cancer. Gallbladder cancer is very much linked to the infection with the liver fluke in the countries where it is common. And you can see that blue countries are the ones where liver fluke can be found. Right? Now, I want to make a case for a couple of countries here. It's rather small. I'm trying to find Ireland. Okay. I don't know if you can see, it's really, really tiny. There's a little Ireland over here. It's an island. There's little Iceland over here. Or is it not? Well, Iceland is somewhere there too. Seriously, on the map, small like this, somewhere there's an Iceland. It's also an island. It's two islands, Ireland and Iceland. In Ireland, sheep herding is extremely common. Guess what? About 25% of population has or used to have liver fluke infection. Just because it's so common. In Iceland, it used to be common too. They got rid of it. How? Say again? Absolutely, yes. That's the only way. They killed all the sheep, and then they waited for some time for the eggs to die in the soil, and then they brought in new, clean sheep. That was pretty brutal but effective way to get rid of that infection. Okay? Now, I want you to, well, on the life cycle, like, where does it go? So you have eggs get in the snail, which is intermediate host. Snail produces, I don't care about terminology, metacercaria, sarcaria, don't care, but from snail, the intermediate form gets on the plants which we consume, and that's how we get infected. And then in humans it develops, we are a definitive host, it develops in the adult worm. Adult worm gets into the bile ducts, stimulates epithelial division, which leads to the gallbladder cancer, practically inevitably, if it's not treated. Ascaris lumbricoides is the largest human nematode. To give you a perspective of how big this worm is, I want to show you this. That's typical Ascaris lumbricoides. It's not getting thicker, okay? It can get longer, up to 2 feet. It's 1 foot. There were species 2 feet long. Okay. Number of cases, 1 billion. That's very famous photograph. The doctor holding Ascaris lumbricoides. This is the obstruction of the large intestine caused by these worms. If you ask about the mass, there were cases recorded where people carried 2 to 3 pounds of worms only. Of course, it's very extremely severe cases. People may have, like kids may have distended bellies. It may really impact the life of a kid. Okay, so it can be life-threatening. Otherwise, if the person has 1, 2, 3 worms, it is usually not felt at all. Okay, so eggs, shed and feces, you get fertilized in the soil, and then when they are fertilized egg is consumed, it develops into the, pretty much passing the larval stage, develops into the larva and adult worm, all in the intestine, no intermediate host. So humans essentially, both intermediate and definitive. Does that make sense? Okay. Now, larva may develop into the adult worm in the intestines, or larvae can penetrate the intestines and get into the surrounding tissues. Now, what is in your intestines except helminths? What other living organisms are in your intestines? Bacteria. Those worms, when they leave your intestines and get into the peritoneum or muscles, do they take a shower? Probably not. So they take all the bacteria, all gram-negatives that are in your intestine with them into the tissues. It can cause all kinds of, from peritonitis to sepsis actually, okay? Disseminated bacterial infection, okay? Now, one of the stages is that larvae go into the lungs. So one of the symptoms of infection is lung inflammation and cough, pneumonia-like cough. From lungs, larvae can go up through the trachea, reach larynx, and you will swallow them, and they will finally develop into the adult worms in the intestine. And they're going to hang out in the intestine for a long period of time, unless you will get sick, like you will develop fever. These worms are very picky about the temperature they develop at. They prefer human body temperature. They don't like it when it's hot. By hot, I mean 104. I mean, if you have a little fluctuation up, nobody cares. But if it becomes hot, they try to leave your body because they think the host is not welcoming anymore. How do you think those worms leave your body, or attempt to do that? Well, there are two doors, back door and front door. Which one do they use? Oh no, these are noble worms. They will use front door. They will crawl against the flow. They will crawl up. So they will go through the stomach and esophagus and the throat, and will try to get out through the mouth or the nose. There are cases, in case of massive infestation, where hundreds of worms at the same time were crawling through the nose, causing suffocation. So it's so massive that, like, if it happens at night, nobody's attending. There were kids that died of suffocation because they just, and you cannot just pull them out. There's so many of them. It's a rare case, but it's pretty horrible, pretty graphic. You can tell, I like to tell very gory details of helminthic invasions. In the United States, helminthic invasions are extremely rare, except for one, which is the pinworm. You've heard about it, right? Pinworm? Heard about it? Every human on Earth has pinworm when they're children. Every person. And then by puberty, something changes and pinworms is gone. I mean, you can treat, but you don't have to. They're going to be gone. Okay. So, which case I wanted to make? This worm is bound to humans. The eggs are shed in the human feces. Okay? And then these eggs are eaten by a human, and you know, they hatch in the larva, and they develop an adult worm in a human, and eggs again are shed in feces. How the heck do the human feces get eaten in, I don't know, by humans on the regular basis? What do we do with human feces? Where do we poop? In the toilet. Where does it go next? Sewage. From sewage, where does it go next? Water treatment plants, right? Some sort of a treatment. Take Africa. Where do they poop? Most of them. Wherever. Or, in some cases, they actually collect feces and use them. For what? Exactly. Fertilizer. Human feces are often used as the fertilizer in developing countries. And there's a bunch of soil-dwelling helminths that are transmitted because eggs are in the human feces. Strongyloides stercoralis, different types of whipworm, different types of hookworms, Ascaris lumbricoides. When you stop using human feces as fertilizer, you disrupt the established life cycle. There is no reinfection anymore. That was shown in places where they switched to the different farming methods, and there you are. Okay. So, we'll talk about the worms. There is not a lot about symptoms that I want you to know, and I will tell you which symptoms kind of, memorable or distinctive ones. Okay, that's number one. And we'll scroll through the study guide. Number two, I will ask about definitive versus intermediate host, but you will not have to write down the entire life cycle, obviously. Does that make sense? So focus on understanding the differences in the life cycle and distinctive infectious features. So I'm asking, for instance, what contributes the most to the transmission of the Ascaris lumbricoides? Usage of human feces as the fertilizer. Does that make sense? Okay. Let's take a break. When we'll come back,